Greetings to all of you. This is the cell structure and function part 2. Previously, we looked at the different simple structure of a plant and animal cell. We saw the prokaryotes. If you've not watched part 1, please go ahead and watch part 1, which talks about even the structure of the cell membrane according to the fluid mosaic model. Now, in this part 2, we are going to look at the different cell membrane-bound organelles that are found within the cell. I remain teacher Dr. Mugomba Ronald, and we are looking at cell structure and function. So we are going to start with the nucleus. The nucleus is a structure that is found within the cell, and it has a nuclear membrane known as the envelope. And this envelope has small pores that allow passage of materials in and out of the nucleus, like the RNAs. The DNA does not go through because it is very big, it cannot cross, but the RNA can cross, the ribosomes can cross after they have been synthesized. Now, the cytoplasm of the nucleus, the would-be cytoplasm of the nucleus is known as the nucleoplasm, and it lies inside the nucleus. It contains the nucleolus, which is usually observed as a dark spherical body in the middle of the nucleus. It is usually where the RNA, the ribosomal RNA usually are formed. The DNA usually forms the chromatins. You will see, when you see under the microscope, you will see the chromatins as euchromatins and heterochromatins. You will look at them and you will see they're usually euchromatins and heterochromatins. They are observed. So the DNA appears as those threads of euchromatins and heterochromatin. You can clearly see this. That is the nucleus. You can clearly see that the outer membrane of the nucleus is continuous with the rough endoplasmic reticulum. This one, we call it the rough endoplasmic reticulum simply because it has ribosomes attached to it. So it qualifies to be called the rough endoplasmic reticulum. You can see the chromatin within the nucleoplasm. The chromatins, some of them are euchromatins and others are called heterochromatins. The nuclear pore is clearly observed. The nuclear envelope has two membranes. You can clearly observe that. So this nucleus is very important when it comes to regulating the different processes that take place within the cell. And on top of that, synthesis of ribosomal RNA from the nucleolus. When you look at the next structure that we are looking at is what we usually call the endoplasmic reticulum. The endoplasmic reticulum is simply a membrane that is bound, membrane bound. They are membrane bound. Structure that is membrane bound. I would say like that. That is the endoplasmic reticulum. Continuous sacs. That is the endoplasmic reticulum. Membrane bound organelle which forms a network of tubes, viscous and cisterna within the eukaryotic cell, except within the red blood cells. Red blood cells do not contain them. But when you look at them clearly, like the rough endoplasmic reticulum has ribosomes attached to it. And those ribosomes are very important sites for the process of protein synthesis. So the smooth endoplasmic reticulum, they are modified like within the skeletal muscle and the cardiac muscle. They are specialized for storage of calcium. Some are specialized for also synthesis of lipids. They are also specialized for synthesis of steroid hormones and also modification of carbohydrates or formation of carbohydrates. Those are the different functions of the smooth endoplasmic reticulum and rough endoplasmic reticulum. So the rough endoplasmic reticulum, because it has ribosomes on it, it enables it to perform a function of protein synthesis, but also allows proteins to undergo modification. So you can clearly see that after the protein has been synthesized, especially those proteins that are synthesized for export, they are not going to be used within the body of the cell. So they are going to be transported to elsewhere. What happens is they are usually synthesized on these ribosomes, which are attached to the endoplasmic reticulum. So after they have been synthesized, they go into the cisterna of the endoplasmic reticulum and they undergo what we call a modification. They work on them very well. They modify them, package them, and finally transport them to the Golgi apparatus. You can clearly see the Golgi apparatus. So it's a network of sacs or network of membranes within the cell membrane. Very important when it comes to packaging. So it's like the post office of the cells. the Golgi apparatus. So the rough endoplasmic reticulum are two, the smooth endoplasmic reticulum and the rough endoplasmic reticulum. Rough is known as rough because it has ribosomes attached to itself outside. The ribosomes are very important for protein synthesis. The smooth endoplasmic reticulum, we said it is very important when it comes to synthesis of lipids and steroid hormones like progesterone and testosterone. They are also very important when it comes to synthesis of cholesterol that is found in the in the cell membrane. Then after there, they package and transport to the Golgi apparatus. So when you look at the Golgi apparatus, it is also flattened big vesicles or sacs known as cisterna, which have two faces. The receiving face that is known as the transcisterna receiving face towards the endoplasmic reticulum, that is known as the transcisterna. So molecules, the vesicles that are coming from the rough endoplasmic reticulum, join the Golgi apparatus at the cis end, at the trans end. Now, as they move from the trans end to the cis end, the molecule, like if it is a protein, it undergoes modification and it is finally released to the cell membrane through the cis end or the releasing phase. The function of this Golgi apparatus is to modify the protein and the lipids and finally transport them to their destination. So it's like a post office of the cells. As you can see it here, you can see this transition electron microgram of a white blood cell that shows visible stacks of semicircular flattened rings in the lower portion of the image. You can clearly see this. That is what we call the Golgi apparatus. Now, after looking at the Golgi apparatus, then we can go ahead and look at another structure known as the lysosome. These lysosomes, the simplest name that you can use to remember them, they are like the suicide bugs. You know suicide bombers, they tie bombs on their bodies and they go to crowds of places and then thereafter they blast themselves and they kill the people around them along with them. So even the lysosomes are very important. They are known as suicide bugs. They are membranous vesicles containing enzymes and low pH. So their role is they are like garbage disposal bags, removing broken organelles and pathogens. The macrophages usually have them. Now, if we are to go ahead and look at the different functions of the lysosomes, when you look at the lysosomes, they are basically divided into two, the primary lysosomes and the secondary lysosomes, as we are going to see. So the functions of lysosomes are basically about 6 of them, and you can be requested to talk about the functions of each of them, the functions of the lysosomes. Now, the first one is they do what we call autophagy. Autophagy is basically breaking down those organelles like a mitochondria that is worn out. That is autophagy. It is going to go ahead and break it down. The lysosomes will come and attach to this mitochondria that is worn out and old. Then they release hydrolytic enzymes that are going to go ahead and digest it all out. Then when you go ahead and look at the second one is heterophagy or cellular digestion. Take an example, the bacterium or the amoeba, or let's say an example of the white blood cell. It can carry out digestion by endocytosis. So when after that digestion, it takes up the molecule. After taking up the molecule, it forms an endosome. Then the endosome, the lysosomes come and bind. After binding onto the endosome, they form the phagolysosome. Then we see digestion taking place of the material. And after the material has been digested, the end products of digestion are used by the cell, and at the same time, the leftover is usually released from the body as excreta by the process known as exocytosis, the body of the cell. Now, the lysosomes are also very important when it comes to autolysis. Autolysis is programmed cell death. When the cells are being programmed, they program themselves to die. So lysosomes are very important for programmed cell death. They release hydrolytic enzymes, and then the cell will be broken down during the due course. Like old cells, they die. and they go on and destroy themselves. It is very important when it comes to development, like the tadpoles, the regression of their tails, that is, even when it comes to different development of the bone, the osteoclasts usually release lysosomal enzymes that remodel the bones. We can look at the lysosomes, their function in fertilization. We can see that the sperm contains hydrolytic enzymes in a structure known as the acrosome that participates in the acrosomal reactions to break down the walls that are surrounding the egg, the zona pellucida and the corona radiata. They are lysosomes are important as well when it comes to the immunity of the cells. They are very important in fostering immunity of the cells. And then also they are very important in the GERL complex. That is, say, the Golgi, the endoplasmic reticulum, and the lysosome system, which regulates the secretory activities of the cell. So this illustration here is simply showing us how the whole process takes place. When the bacterium has swallowed the food by endocytosis, a vesicle is formed known as the phagosome or endosome. Then the lysosome comes as indicated and then binds to the phagosome to form the phagolysosome. Then hydrolysis of the food takes place. After the end products of digestion are released into the cytoplasm. What remains is usually released or ejected out through a process known as exocytosis. Now, we can go ahead and look at this system known as the endomembrane system. It's a system of different structures, different shapes made up of membranes, and their role is very important when it comes to modifying proteins and lipids. As we have seen, it all begins with the cell membrane, the envelope, nuclear envelope, the rough endoplasmic reticulum, the smooth endoplasmic reticulum, and the Golgi apparatus, along with the lysosomes. They play a very important role when it comes to making and modifying the different proteins. So you can see that relationship. It is very clearly indicated here. So it all begins with the nucleus. Across the nuclear envelope, the information goes being carried in a form known as a transcript known as the messenger RNA, goes to the ribosomes, which are found on the rough endoplasmic reticulum. A protein is made, then it is packed in a vesicle that goes to the cis face of the Golgi apparatus. Then it undergoes modification and folding. Then it is released at the trans end of the Golgi apparatus. Then thereafter we can see clearly that it is excreted through the cell membrane. So that is the system that we are talking about. Now we go ahead and look at our next organelle known as the lysosome. Lysosome is very, very important. They are organelles which are they are not made up of any membrane. They are organelles not made of membranes. They made of the RNA and protein. They just have an RNA strand that was formed from the nucleus and a protein. So usually they usually attach to the rough endoplasmic reticulum, and their function is for making protein synthesis of proteins. Now, when we go ahead and look at the powerhouse known as the mitochondria is very important. It is like the powerhouse of the cells. That is where energy is formed. That is where we take chemical energy. So that's where we take our food that we eat and we break it down to form ATP. And the ATP is used to sustain different processes within the body. When you look at the structure of the mitochondria, you will be surprised to see that the mitochondria is oval-shaped, though the shape of the mitochondria varies from species to species. You'll find some organisms having spherical mitochondria, but the most commonest is the oval-shaped mitochondria. It's a double membrane organelle. That means that it has the outer membrane that is usually smooth. It is usually smooth, and the inner membrane, which is usually folded to form cristae. The cristae are very important when it comes to increasing surface area over which energy production takes place. Between these two membranes, usually we have what we call the intermembrane space. Intermembrane space is that space between the inner membrane of the mitochondria and the outer membrane of the mitochondria. So the liquid part of the mitochondria that is enclosed by the membranes is known as the matrix. This matrix here is the one that usually contains the hydrolytic enzymes, the enzymes that participate in a process of metabolism known as Krebs cycle. In Krebs cycle, we have a number of enzymes, a host of enzymes. So all those enzymes are usually found within the matrix. So you can clearly see here that those foldings that are found on the inner membrane of the mitochondria, those are the ones we usually call the crysts. And they are very important when it comes to providing a larger surface area for energy production. They are very important. The matrix usually contains, mitochondrial matrix contains the enzymes which participate in the Krebs cycle. The outer membrane is usually smooth, but it is usually permeable, freely permeable, so that it can allow oxygen to enter the mitochondria and allow carbon dioxide to get out, because carbon dioxide is a product from the process of respiration. Having looked at the mitochondria keenly, you should be in position to talk about its structure. You should be in position to talk about how it is adapted for its function and the function that it performs. Now, we can go ahead and look at the chloroplasts. The chloroplasts are viscous with two membranes, like the mitochondria also. The chloroplasts are also two double membrane organelles. And for these ones, for them, their role is to carry out photosynthesis. Photosynthesis is basically that process that plant cells use to synthesize food materials using sunlight energy, carbon dioxide, and water to synthesize carbohydrates, of which those carbohydrates will be converted to the different forms of foods. When you look at the chloroplast has an inner and outer membrane, which are both smooth. They are both smooth and selectively permeable to allow certain molecules to enter the chloroplast and restrict others from entering. When you see the chloroplast, as we are going to see in the image that will come thereafter, you can clearly see that within the liquid part of the chloroplast, that liquid part of the chloroplast that you can see, it is known as the stroma, this one here. It is very important because it is the one that contains a host of enzymes which participate in the process of photosynthesis, especially the dark or the light-independent state of photosynthesis takes place in the stroma. So that is where we find those enzymes there. When you see within the stroma, you realize that there are stacks of coin-like structures. Those stacks of coin-like structures are known as the granum. One of them is called a granum, and then each of those coin-like structures are known as thylakoids. Those thylakoids are fully packed with a green pigment known as chlorophyll. Chlorophyll pigment is very important when it comes to trapping sunlight energy and ensuring that we have enough energy to carry out the process of photosynthesis. So, the light-dependent reactions of photosynthesis usually take place within the thylakoids. So you can see this one also has an intermembrane space between the inner membrane and the outer membrane. That is very important when it comes to synthesis of energy. We shall learn about the process of synthesis of energy by a process known as chemiosmotic theory of ATP production. Chemiosmotic theory of ATP production is very important. That is where we talk about how ATP is generated as a result of a proton gradient that is established. So thank you so much for listening. We have been looking at the chloroplast in this part 2 of the cell structure and function. We looked at the chloroplast, we looked at the mitochondria, we looked at ribosomes, we looked at lysosomes, we looked at also the rough endoplasmic reticulum, the smooth endoplasmic reticulum, in details, each of them, how their structure relates to their function. Thank you so much for hanging on up to now. I remain teacher Dr. Mugomba Ronald. Remember to subscribe, share, and comment. Please like, Share with your colleagues when you find it useful. I remain teacher Dr. Mgomba Ronald. Have a good time. Greetings to you all. Till next time.